Hello. Now we're going to do Lab 2, which is going to focus on the microscopes and cells. This is in the Sarakas exercise lab book. It'll be exercise 2 and 3. So let's get started. First off, what you should be doing is pre-reading the exercise 2 and exercise 3. Now it's marked CH2 and CH3, but it basically is the same thing. Review the microscope structure and principles that we're going to do. Complete the review sheet. Now the review sheet is at the end of the exercise book section, but you don't have to do that. I've included the sheet in a PDF format, which you can easily fill in each of the questions and then save it as a PDF. So you will do exercise 2 for the microscope and exercise 3 for cell and cell structure and cell division. You need to upload those at the Blackboard site, and it has to be done prior to Friday, 5 p.m. I do not take late work for this week. You want to review page 16 and know the parts of the microscope. Now, this is very important because what happens a lot of times is that students spend a lot of time filling out sort of the worksheet part and ignore everything else. Don't do that. One of the things that you're going to find is that there's going to be certain questions that you'll have to answer that are not in the worksheet, but that are actually in the exercise pages. Please fill those out in all cases because you're going to be able to use this material to review prior to your lab practical exam. What you also need to review is how to handle and pick up the microscope. Now, you'll be helped with this if you go to the Blackboard and for the section of lab videos, you review those videos. Some of them are relatively short, but some of them are very detailed, and they'll show you how to properly handle and pick up a microscope. You need to be able to know what is the coarse adjustment knob. If you were to do this by hand in a lab, you would first, of course, put the slide onto the staging and use the lowest setting and use the coarse adjustment knob first. You get an image that's somewhat there, but if you want really fine detail, then you begin to act on and handle the fine adjustment knob until you get a good, clear, final viewing of the specimen. Here, of course, are the eyepieces. Yes, you need to know the anatomy of the microscope. You may have done this many times before or very few times before. One of the things that happens nowadays is most microscopes will have this larger, wider knob here, which is the coarse adjustment, and then you just have to move your fingers slightly for the fine adjustment, which is on the inside ring. The illumination is always coming up through the stage. There is a condenser, which allows you to modulate the amount of light going through. This is a mechanical stage, so you'd put the slide into sort of the gripping structures of the mechanical stage and be able to smoothly, by the stage control knobs here, move the slide. But remember that everything is inverted. So when you feel like you're moving the slide upward, it's actually going to appear that you're moving the slide downward, and vice versa. If you feel that you're moving it left, it'll appear, because of the lens system, that you're moving the slide right. The nose piece contains several different objective lenses. The objective lenses are set up in such a way that there'll be a short stubby one. It's usually a 4x. So you take the magnification of the short stubby objective and you basically multiply it by the ocular lens. The ocular lens, on average, there are a few exceptions, but most of the cases for a standard laboratory format is that the magnification for the ocular lens is 10x. So 10x times the objective of 4 gives you a magnification of 40. Don't panic as much because of the fact that, gee, do I got to remember every little detail? One of the best details to save you from a lot of problems is first and foremost, use the shortest objective lens first. You might not see as much, but as you increase in magnifications, flipping the nose piece to the next objective lens, you'll see greater enhancements in magnification. What happens in this head area is a series of prisms and lenses that are able to take and guide the light up through at an angle into your eyepieces. Okay? This is the frame, this is the arm, and this is the base. How you handle a microscope, by the way, is by grabbing onto the arm and cradling underneath your forearm, or on top of your forearm, I should say, excuse me, the base. So you have a firm handle. Never grab the eyepieces because they'll pop out. And then you would end up dropping the entire microscope. And that can be $400 or $500 of tears. Well, anyways, this is the mechanical stage that I'm talking about. So the slide would be put into this little area here. It allows you to smoothly move the slide left to right, forward, and backwards. Okay? A lot of times now you don't have the knobs back here, but you'll have them underneath. Here you can see the 4, that's the shortest of the objectives. So this would give you a 4x magnification. There would be other ones usually that go 10x and then 40. And so 10x times your eyepiece 10x would give you 100x magnification. 40x times the 10x gives you a 400 magnification. For most things that are eukaryotic, have a nucleus, you're not going to need more than a magnification of 400. This bigger, larger one is only used in oil immersion for microbiology viewing because it would give you a magnification of about 1,000, and that requires special arrangements, but you won't need them for this class. Here's a more sophisticated arrangement for the mechanical stage. You can see where the translational control knobs are. You would be moving the slide back and forth here or up and down. So basically in the Y position or in the X position. Okay? Now, if you move forward from here, I've already talked about this a minute ago. If the microscope has a 10x eyepiece and you have a 10x or 40x objective, what you have is a magnification that's basically a product or a multiple of both the 4x and the 10x, which would give you 40x, the 10x and the 10x, which give you 100 magnification, and the 40x and the 10x, which give you 400. Working distance. Oh, that is so important. Why? This is the distance between one of the objective lenses and the microscope stage, or really the top of the slide. Why is this so necessary? Because you create a hazard if you're not attentive to the working distance of broken glass. Now, if that was a preserved specimen, let's say it was in a pathology lab, and you had a sample, you could contaminate the stage with the broken slide and the material that was inside of it. So we have to get used to certain terms. One of them is working distance. The next one is the field of view. This is the illuminated area that is observed through the eyepiece. Now, sometimes too much light can be a disadvantage. You can wash out or basically make very hard to see some fine details. That's why you would use the adjustment iris to control the light passing through the specimen. Okay? The field of view obviously will be decreased as you increase your magnification. Why? Because you're going to be having more a focused area on just a smaller subsection of that bigger view. All right? Parfocal. This is great if you have a properly adjusted microscope. Most microscopes in the labs that you have are usually they're not really sent out, but they bring in a microscope um, adjuster and maintenance program that occurs every summer. They basically grease the gears, remove any broken down gears, remove any uh, broken lenses, make sure that everything is in proper alignment so that you can create the condition of parafocal. What is it? Let's say you are having sets of objectives or eyepieces that are so mounted that they may be interchanged without varying the focus of the instrument. In essence, if I go from 40x to 100x, just as I flip on the nose piece from one objective to the other, I should have everything pretty much still in focus. I won't have to adjust much. And if I do, it's just a little bit. But parafocal is really incredible when you think about it. You don't have to redo all the magnification. It's as if you're just getting a more focused and smaller area view on a particular detail that you're trying to study. Here's an example of working distance. If you can see here with the magnification for, I believe that's 10, and this one is 40. Notice the difference, because what happens is that for the greater magnification, your objective is going to be longer and longer. As a result, your working distance between the slide and the tip of the objective, which is where the first lens is, becomes less and less. That's why it's so important, A, to start off with the smallest lens, the shortest one, which would be the 4x. What you want to avoid is a condition that happens sometimes when you flip the objective. You're not attentive to what's going on, and this, literally, the tip of the objective crashes into the slide. It will break the slide. You may damage, scratch, or actually break the lens inside here. And it can be also a possible route of contamination of the specimen that's here, now spreading contamination onto the stage. Now, this is what I was talking about, the field of view. At 40x, here's my structure. I can't really tell much, but if I increase my magnification, I can see it looks, hey, almost like a rectangle. It's got more view. But if I go 4 times that to a 400x, notice this. It's somewhat rectangle, but on each of the corners, they're somewhat cut off. I've increased my view, even though my particular area here from this side to this side has decreased. I've focused in on one particular aspect, and I have increased my magnification. So my field of view decreases while I increase my magnification. Parfocal. Here's an example of this where you see this. In low power, you can see a variety of these cells, and they're all over the place, etc. But if I push this to a higher power by changing one objective to another, notice that I get to see more detail in a cluster of these cells. That's what you want. Now, there's a principle of inversion of an image. As remember I mentioned earlier that if I move the slide, left seems right, up seems down, right seems left. That's because of the images that pass through the lenses. Inversion occurs as the image is reversed by being passed through the lenses of the microscope. So what starts off, and usually in training in a lab, you would get one of your slides and it's marked just with a typewritten letter E. But as I put this under the microscope, you're going to notice something. First, the E becomes upside down. That's the inversion. As I increase in my power, I get to see more finer detail here. This is from newspaper. And you notice eventually the small fibers that make up the newspaper as you get into higher power. But the inversion turns the item upside down or backwards. But as you increase your power, you can see more detail. So it doesn't really matter in a way. The principle of inversion of image is because as light moves from air and then changes into glass, it tends to cause a curvature change. This is as you're building up a focal point here. through a convex lens. Now, one of the things, notice the specimen. Where is the flagella here? Where is the nucleus? This is an euglena, by the way. But notice that as it's passed through, you see instead it is inverted, reversed, and enlarged, which therefore you're going to get more detail. But the image is going to be basically the bottom is now on the top and the top is now on the bottom. Depth of field. This refers to the thickness, the depth of the tissue layer that is currently in focus. Now, what you have to keep in mind is this. Even if I take a very, very thin layer of cells, a tissue, for example, muscle tissue could be anything, and I place it on the glass slide, it has a level of thickness. As you increase your magnification, you're going to notice differences in where the actual focal point is. It's going to become much more pronounced as you increase your magnification. So you need to be aware of what the depth of field is. The best use or best demonstration of depth of field is a slide that a lot of times is used in this beginner's part of microscope use, and it's called colored threads. What you have are 3 threads, yellow, red, and blue. And threads are actually made up of literally smaller fibers. They're all stained at a particular color, but they're all laid on top of each other. So they basically have a differing depth of field. The same thing occurs if you've ever watched news items and it's like live at the scene of the fire or crime or whatever. If you notice, the cameraman sets the focus right on the face of the reporter. Behind that reporter is slightly out of focus. So the focal point is really on the reporter. If the cameraman has to turn over as the reporter says, well, okay, if you look over on the side here, there's this occurring, he will have to adjust the focus so that the viewers can see clearly a distance away that particular event that the reporter is referring to. Now, depth of focus has been defined as the vertical distance that an object remains in focus at one time. So if you notice here, this seems to be the most in focus, then it's next, and then you have the one that's further down. If you focus on one thread, the others will be out of focus. So if I shift it over to the middle one here, this would be out of focus and this would be out of focus. And this occurs all the time. You have the field of view here, and this slide gives you some idea. Some people also referring to this as calibrating your microscope, okay? But it allows you to look and see images. And one of the things you got to keep in mind is, let's say something like a cell here. You're going to have to move that focal point through the depth of the cells. Some parts of it will be in focus, others will be out of focus, whether they're literally above or below that field of focus. And what I mean by out of focus is that something will appear somewhat blurry looking. Now, determining the diameter of the field of view, this is not hard. Remember we talked about this a few minutes ago when we moved from 40x to 400x. Okay, but if you look at a lot of the microscopes, at least one of the eyepieces may have a grid on it. And this grid, you'll be able to calibrate. For example, 1, 2, 3, 4. You have 4 millimeter diameter. A millimeter, which is milli means 1,000. Okay, so 1,000th of a meter. You can break that down further. And when you get into micrometers, micro refers to millionth of a meter. So 1 millimeter here is equal to 1,000 micrometers. So if you have something that's, let's say, halfway across, it would be 0.5 millimeters, or basically 500 micrometers. And that's what you have in using the grid to get some idea about things like the cells, how big they are, how they span across a view, and how as you increase your magnification, you're going to get to have a reduced field of view, because remember, your field of view will decrease as you increase your magnification. Oh, and by the way, micrometers, another way to have a shorthand for it, is sort of like this inverted Greek symbol. It looks like a U backwards. Now, you have here your field of view. As you can see here, this is your area of view, and you're going to see how these change as you go from a 40x to a 400x. Now, from there, we've gone through the microscope part of the exercise. Now we're going to go into the cell structure and cell division. Do remember that there are some videos that I've put aside, and you are required to view them. They will be helpful in your understanding of this lab exercise, as well as helpful in understanding some of the basics of things like the cell structure, etc., mitosis, etc. You should review the cell organelle structure and functions on page 28 of the Sarakas lab exercise text. A basic cell will consist of a nucleus, a cytoplasm, and a cell membrane. Now, inside of any eukaryotic cell, and all eukaryotic cells have a nucleus, membrane-bound nucleus now, you're going to find also membranous organelles and non-membranous organelles. Another way to say it is organelles that have at least one membrane containing their biochemical activity inside, and other organelles that have no membrane at all. When we look at membranous organelles, these include the peroxisomes, lysosomes, Golgi apparatus, mitochondria, which actually has two membranes. And really, it's not as much a membranous organelle, but you're going to find from time to time free ribosomes. But the thing is, some of these ribosomes are not going to be not only not containing organelles, or I should say, they're not membranous, but some are not free and they're actually attached to a membranous organelle called the rough endoplasmic reticulum. In the rough endoplasmic reticulum, they're studded with, basically covered on the exterior with ribosomes. Let's go over a few other points. Peroxisomes are basically a membrane-bound structure that is going to be involved with basically breaking down or neutralizing certain reactive oxygen species or free radicals. Lysosomes are going to be basically a membrane-bound structure that contains digestive enzymes. They will break down other types of proteins and other materials. The Golgi apparatus is sort of like a shipping department. It takes in vesicles, which are membrane-bound structures that contain various types of materials, such as proteins, etc. It will purify, package them, and chemically process them, and then release them in vesicles, whether it's to be shipped outside the cell or to other places inside of the cell. The mitochondria is the unique organelle that makes ATP molecules. And it does this through some very sophisticated processes that include the citric acid cycle and the electron transport chain. Ribosomes are the protein synthesis structures. They are involved with taking the genetic information transferred over to them by a long molecule of messenger RNA, transfer that information into a format of a line or assembly, a chain of amino acids, okay, basically involved with protein synthesis. Now, I talked about the endoplasmic reticulum. What happens with rough endoplasmic reticulum is that the ribosomes are still making proteins, but they inject them to the inside of the endoplasmic reticulum, the rough endoplasmic reticulum, where they may be adding various sugar groups to them, and they are then eventually packaged and sent out in vesicles to eventually be processed further by such structures as the Golgi apparatus. Okay, that sounds nice, but what about the smooth endoplasmic reticulum? Yes, its outer surface is smooth, but it is extremely important as a membranous organelle. Smooth endoplasmic reticulum is involved with a variety of chemical reactions. And those cells that are involved with very sophisticated types of chemical processing, etc., such as the liver, or in some slight lesser cases, the kidneys, they're going to tend to have lots and lots of smooth endoplasmic reticulum inside of their cells. Smooth endoplasmic reticulum is involved with um, lipid synthesis. It is involved with certain breakdown of certain toxic chemicals, alcohol, certain drugs, etc. And it's also involved with maintaining certain levels of glycogen structures. And basically, it's a very interesting area. You can take the liver cells from a teetotaler, someone who has never drank ever any alcohol, and someone who's a heavy pounder of alcohol. And you can literally take tissues, stain them for the smooth endoplasmic reticulum, and you'll see great differences between one person and the other. The person that pounds down the liquor all their life, they're going to have lots and lots of smooth endoplasmic reticulum in those liver cells. The teetotaler is going to have much, much less. Okay? Now, in the nucleus, this is a membrane-bound structure that you can find several important points, some important structures. The nucleolus, which is a sort of darkened staining area within the nucleus that is involved with the manufacture of ribosomes. It also makes certain histone proteins. It's involved also in the processing and contains a lot of RNA. Now, chromatin, that is the DNA. It may be wound tight or kind of loosely extended, and that's where you're going to have, of course, the first part of DNA reading that will eventually lead to the synthesis of messenger RNA, and it'll be sent outside of the nucleus. The nuclear envelope itself is actually double layered, and one of the interesting things is it's also going to have little pockets or holes in it. They're called nuclear pores that allow certain molecules to go in and out. Non-membranous organelles. Well, these include the cytoskeleton. There are three major types. Microtubules, intermediate filaments, and microfilaments. These are all made of different types of proteins and are involved with structure as well as various types of intracellular transportation. And in some cases, some of the components are essential for some cells that do things like contraction or transport of materials down long extensions of the cell, such as the axon of a neuron. Centrioles are clusters that contain microtubules, and they may be involved with forming the spindle fibers that are part and parcel of the mitotic process. Microvilli are also held up as extensions of the cellular membrane, and they're supported by microfilaments. And then we have, of course, the plasma membrane itself. The plasma membrane is made up of phospholipids. It's a double membrane structure that basically acts as a barrier for materials coming from the outside of the cell into the cell and from the inside of the cell out of the cell. It restricts the movement of various ions, water, wastes, etc. It's primarily composed of glycolipids as well as phospholipids. There is some rigidity that's added on by the inclusion of cholesterol, and we'll find out more. It is the cell membrane, sort of the barrier that helps contain the cell. Okay? If you look at this diagram, you will see this. Here you have the microvilli here. You have secretory vesicles. Some of these vesicles will fuse to the edge of the cell and release their cargo to the exterior of the cell. You have the nucleus here. The nucleolus is in the center here. There is also chromatin here. Chromatin is basically composed of DNA. You have your cilia as well present, and you have also proteasomes, which are structures that help in rearranging, recycling, or destroying of abnormally folded proteins. Here we have ribosomes, which are basically RNA, ribosomal RNA, and proteins that come together and form these functional structures for synthesizing proteins. And we've talked about peroxisomes that contain degradative enzymes to reduce the presence of reactive oxygen species, free radicals as they call them. Lysosomes, which are present here, that contain various digestive enzymes. You will also see the plasma membrane, and also the plasma membrane has a variety of proteins in it that allow sort of like channels for certain molecules, certain ions, etc., to pass from one side of the cell to the other side. Cytoskeletal elements, we've talked about centrosomes, centrioles, and you've had also some of the membrane-bound structures such as the Golgi apparatus, the mitochondria, and the endoplasmic reticulum, both smooth and rough. Okay? So I encourage you to review these and get these familiarized. One of the points to keep in mind is that labs, whether they come before or after the lecture, are used to help enhance the comprehension and knowledge that is taught in the lectures. As we go through our cell membrane, we mentioned before, it consists of a phospholipid bilayer. Bilayer meaning two. There's two layers. Both the layers, the hydrophobic ends, face each other inward because the hydrophilic, the water-loving sides, will be focused either on the exterior of the cell or on the interior, inside the cytoplasm of the cell. The cell membrane is a semi-permeable membrane. Some materials can pass through, some cannot. Cholesterol, as I mentioned, adds rigidity to the membrane. It also has the presence of glycolipids. There are some glycoproteins present too, but we also find that there are proteins. The integral ones pass from one side to the other. These include enzymes, carriers, such as channels, and some ion carriers, etc. And then we have peripheral proteins. Some of them hang on the outside or the inside layer of the membrane, but they don't pass across totally. Here you see a good example of this. Outside here is the extracellular fluid. Inside here is the cytoplasm. Remember that there's a presence of water on both sides. Okay? Here we have some of the cytoskeletal, the microfilaments that provide structure, etc. Here we have some of the peripheral proteins that hang on the interior. Some of them may be on the exterior. Integral glycoproteins. Glyco means that they have some sugars attached. And as you can see here, here's one that's integral and has a chain of sugars, carbohydrates on the outside. We have integral proteins that may have an interior that allows for the passage of ions, water, certain ones allow for the passage and the transport of glucose from one side to the other. Some are specialized. They're gated channels that do not allow, unless they're stimulated, the passage of ions, etc. through. This is where we get into the phospholipid bilayer. Remember that the beads, the red heads here, these are the polar part, maybe phospho with several other components. And the tails here are of fatty acids, which are nonpolar, they're hydrophobic. The heads are hydrophilic, water-loving. So you're going to have this boundary here that's hydrophilic, this boundary here that's hydrophilic, but inside here is nonpolar, hydrophobic. So molecules like cholesterol, which you see here, which enhance the stiffening of the membrane instead of allowing it to flow as freely, as well as the fatty acid tails will be down here. All right? Now, Normally, this would be a process that you would be doing some laboratory activity. Unfortunately, we don't have that capability. If you were to make a wet mount, you would probably be using a simple dye like methylene blue or something to that effect. Um, in all labs that you do hands-on, you would have certain requirements to be aware of. The lab biohazard as well as the general hazard. General hazard would be you're going to watch out for using the staining from the dye. You could end up stained on your fingers and it stays there for some weeks until the skin cells slough off. You also have to deal with the fact that you're dealing with slides, etc., that you have the risk of a broken glass and a cut from that. Now, we're not going to be doing this, but if you were to do this, and you could do this with a simple scraping of your cheek cells inside with a toothpick, and then you just basically smear it on the slide, add a drop of water or a drop of stain, add a cover slip, which is also usually a very thin piece of glass. That's why it's also important to be mindful of that. And then you make yourself basically a wet mount slide that you can then peer at and look at some of these cheek cells. The biohazard issue has gotten more prominent because usually we don't want to say it, but we want to basically recognize that anybody and everybody has the potential for some communicable disease, etc. So usually after the student has made their slide, they dispose of both the toothpick and the slide in the proper biohazard container. Okay? Now, here are the basic directions on making a wet mount. And you could do this for wool, uh, some hair, an onion skin, cheek cells, etc. Whatever specimen you use, you basically have the clear cover slip. Now, some of them are also made of plastic, by the way. And it comes up to the edge of the fluid, and then you drop it simply down. You should be able to evacuate and not have any air bubbles here. And you lower slowly, and it becomes nice and smooth. It holds the cells, and they are going to be stained with that certain blue stain. You want to make notes of observed slides. Well, we won't be doing that as much, but I wanted to bring up another area that's important. What are the differences and limitations of light microscope versus electron microscope? Some of this is discussed in Sarakas, the author's lab book. Basically, when you think about it, a light microscope was what we had from the beginning of the 1600s forward. But there is a magnification limit, and that's usually about 2000x. So 2000 times magnification, that's about the limit. But when we got into the world of dealing with viruses and things, they're much, much smaller. And so, even though viruses were discovered in basically the late 1890s, nobody had really seen what a virus looked like, although they had a lot of experiments that demonstrated the presence of them until the onset of the electron microscope. The electron microscope finally showed the appearance of viruses, and that was around 1935. Incredible when you think about it. Because basically, when you look at it, there's about 30, almost 40 years where they had the existence of something, but they could never see it because it was too small. You see, electron microscopes, whether you have one that scans on the exterior of something or can transmit and penetrate into a structure, can see far smaller structures. In certain EM images, you can get magnifications of 100,000 times X, 200,000 times X, yes, even up to 500,000 times X. That's because the wavelength of an electron is far smaller than the wavelength of light. There are some limits, though. Light microscope, you can see things alive as they move around. You can make video. You can collect images in color. Electron microscope requires several important factors when you're taking the image. One, the image has to be either frozen or dead. And if it's frozen, it's probably dead anyways. Two, the images do not come with color. They're black and white and grayscale. You do see today color images, but that's using a computer to artificially add color, especially to emphasize certain structures compared to other structures. You do not get video. Sorry, can't see things moving around. And so that's a frustration, because an electron microscope, you have to put the sample into a vacuum. So there are no gas molecules to bump against the electrons as they're coming down to basically reflect off of the image, okay? And as a result, that they're in a vacuum, and they may actually be frozen to prevent movement or anything, you get a great microscopic image, far more intense than you would with a light microscope, but the object is dead. As you can see here, here are some of the comparisons. Visible light as their radiation to see with a light microscope. With the electron microscope, it uses an electron beam wavelength. And that compartment that you see right there, that green area, that's all pumped out. And it's a vacuum, because any gas molecules, as I said, would make the imagery blurred or impossible, because that's where the electron beams would bounce off of instead of bouncing off the target. Here's the difference when you look at images. This is a light microscope. You can see the chloroplast moving around in the cells. Here is pollen. This is with an electron microscope. You get far more detail in these small images. Look at the spikes, etc., on the different types of pollen, etc. But no movement, and everything here is in a vacuum, and everything here is usually covered, or sputtered as they call it, with some type of coating that allows the electrons to bounce off of them so they get a good surface image. It could be atoms of osmium or tungsten or gold. But the idea being is this: you're not going to get motion, you're not going to get movement, you can get finer detail, but no video, no movement, etc. And also notice this is all sort of a gray scale here. There's no color. So let's compare them quickly. Light microscope, less resolution; electron, more resolution. Light microscope, less magnification; electron microscope, more magnification. Light microscope, easy to carry; electron microscope, cumbersome. It is a desk. It is a large piece of instrumentation you would see. Light microscope, color images; electron microscope, black and white only. Finally, we're going to go through mitosis. Now, we have to go through the cell life cycle here, and I encourage you to review the pages of 34, 35. You want to review the pages, the phases of interphase, and basically how all these phases will contribute to preparation for cell separation, cell division that we call mitosis. Okay? You want to review the steps of mitosis: PMET, prophase, metaphase, anaphase, telophase. Observe slides and locate examples of each stage of mitosis. Now, in the cases where you don't have slides available to you, no sweat. Look at some of the video imagery that I've provided for you in some of the associated videos. That should help you. If you take a look at interphase, interphase is sort of this part of the life cycle that's not part of mitosis. So interphase, once you've separated into two cells, you go into sort of this G1, or it'll go into sort of G0. Think of G0 as if you're running the car, you've probably done it in the morning, and the engine is idling, and you're just doing it to warm up the car. G0 is the cell literally can put its feet up, it maintains certain biochemical activity, but it's not planning on having any further cell division. But there are certain steps that will get the cell moving forward. Now, during G1, you've now separated from one cell into two daughter cells. So what that means is that you're going to have normal cell functions plus some cell growth, perhaps making more organelles as necessary, and having protein synthesis. When you eventually get to sort of a checkpoint, you then go into S. Once you've gone into S, that's it. You're committed because why? S is where you have DNA replication. You're also going to do a lot of synthesis of histones. Now, let me explain to you. Think about this. If you take a look at your cell, any one of your cells, you have basically 46 chromosomes. In other words, you have 23 pairs. If you took each one of those, unwound every one of those chromosomes, so they're just a long string of DNA, you'd have DNA about the length of about 2 meters, a little over 6 feet. It's a lot of DNA. And you have to basically duplicate that. So you now have, instead of 46 chromosomes worth of DNA, you have 92. How do you keep them separated? How do you keep them ready to be divided? Well, that's going to happen in a little bit when we start getting into mitosis. But what we have to do, first and foremost, is replicate all of that DNA. And what you also have to do, and that's why it takes 6 to 9 hours or 6 to 8 hours, as it says, you have to make sure that you have no errors. And in that proofreading, that error correction, you have to do that also during that process. Now, in the end, the cell has double the DNA. It moves to the next section, which is G2. Now, G1 as well as G0, S and G2 are all phases that are part of interphase. Don't get that mixed up with mitosis. G2, you have to go through some final stages of preparation. And in this case, you're going to do a lot of protein synthesis, particularly for a protein called tubulin, which is necessary to make the spindle fibers, etc., to separate out the chromosomes later on when you go into mitosis. Once you get to the point where you're at this checkpoint here, you start mitosis. Mitosis breaks up into prophase, metaphase, anaphase, telophase. There is a video, at least one of them, that shows the separation of these cells going through mitosis, and I want you to view that because we can tell you a lot of and show you a lot of static pictures. It's just still pictures, but the incredible activity of actually seeing the separation, where basically you have a cell that has all its DNA contained in the nucleus, the nuclear membrane breaks down, the formation of the spindle fibers, the lining up of all the chromosomes in metaphase, the pulling apart of the pairing of the chromosomes during the anaphase, and eventually the repackaging and the separation of both daughter cells during telophase. That is incredible. Also, one other thing. There's this little term here down called cytokinesis. Cyto is cells, kinesis separation. Cytokinesis, depending on the type of cell, it may start even as part of anaphase, or it may be occurring during or at the end of telophase. It totally depends on what type of cells you're talking about. Muscle cell, kidney cell, liver cell, skin cell, etc. They all have slightly differences in when they initiate cytokinesis. But this actually got to be confusing for some people because when they read some of the older biology books, cytokinesis would be sort of the fifth state after telophase. But now they recognize that that cytokinesis can occur anytime, even in the midst of anaphase. So when you go through mitosis and cytokinesis, you're going to see these stages: early prophase, late prophase, metaphase, anaphase, telophase. Now, when telophase is occurring, you have this unique separation that occurs, and it's called a cleavage furrow. It's almost as if you remember those string purses. You pull on the strings, and the opening to the purse closes up. Same thing if you have a hoodie and you pull on the strings, what happens? Well, the front of the hoodie kind of closes up. It's great to protect you when the winds are cold, etc. But the cleavage furrow is sort of a ring made up of actin, myosin, etc. And what's going on is they're just sort of this pinching process to separate out the two daughter cells. Okay? You want to see that the centrioles play this role initiating the formation of what's called the spindle fibers. The spindle fibers are going to basically trap and set up the arrangement of what's called the equator here. This is during the metaphase plate. You take the paired chromosomes, you line them all up so that one of the pairs of the chromosome goes north, one goes south, etc. This is occurring as the separation occurs during anaphase. Well, now that I've covered all this, here's what I want you to do. I want you to review chapter 2, review the videos on handling the microscope and the parts of the microscope, etc. Complete whatever parts of the exercise 2 of the Syracuse in your lab text that you can, as well as the handout sheet, the worksheet, which is pages 25 and 26. You're going to upload those to the Blackboard site. Do not hand them to me. Do not email them to me. Anything that you email to me as an attachment, I delete. Now, that's not to be mean. You have to understand why. A lot of things can be sent, including viruses and Trojans, by attachments. Furthermore, and this is the other very important point, if you send it to me directly by email, I can't grade it. If you upload it at the Blackboard site, I can grade it, and you can see what the grade is afterwards and see where you did well, where you may not have done as well, etc. And that's the important point. I've got a place where there's hard evidence, where you have done a great job, or maybe where you need to polish up some things. This same applies to the exercise 3, chapter 3. You're to do all the exercises 3.1, 3.2, 3.3, 3.4. Fill those out in the pages because you can use those to review to prepare for the lab practical. You're going to take the handout that I have in PDF format at the Blackboard site, download it, complete it, and then upload it again. Remember that after you've made all your alterations and saved all your information and done the proper writing of the answers, etc., that you've still got to save it as PDF to upload it. You cannot send me a document in Pages or any other open source software, because what happens is Blackboard will not accept it. It'll be something I cannot read, and therefore you get a 0. Okay? Do take the time to review the lab videos, not only for this week's lab, but then for the next one going forward, because the next lab is going to be very soon, and that's going to be involving exercises 5.1 to 5.5 as well as 6. Well, have a wonderful day.